This is the insulin presentation for cell biology by Jake Thompson. To start off, there is a summarization of proteins in general. Proteins, they have 4 levels of structure. They have primary, secondary, tertiary, and quaternary. This is an example of a tertiary structure. And you can see that there's disulfide bonds, but you cannot see the alpha helixes and the beta sheets. Proteins can function in multiple ways. They can function as hormones, enzymes, and these 2 primary ones are the 2 main functions of the insulin protein. Insulin can also help with the transport of glucose across the cell membrane. It can also help transport other things throughout the cell, but it's mainly glucose. Excess glucose that is gone through can be stored as glycogen, which is made by insulin. And if you do not store all this excess glucose, you can have hyperglycemia. And if you didn't have enough, you can also get hypoglycemia, both of which can have drastic consequences on the body in both the somatic and the autonomic nervous systems. So you can see here the sequencing of the insulin protein. It has 51 total amino acids, but it has a first chain known as chain A and a second chain known as chain B. And whenever it is made from proinsulin, which is what insulin comes from, it actually has 81, but the C polypeptide, or just connecting polypeptide, it cleaves off these 30 extra. These 30 extra will go on and move away from the cell, and it'll allow the insulin to form the hexamer to do its processes. Insulin is mainly found in animal cells, but there's actually been evidence to show that it is found in prokaryotic cells, protists. It even has several fungi that have these. And this is actually kind of important because we extract all the insulin that we use commercially. Like whenever we extract from yeast, that's one of the main sources. You also use recombinant DNA and bacteria, and this allows for the production synthetically. It can help people with diabetes mellitus with type 1 or type 2. So the INS gene encodes the proinsulin, again, which leads to normal insulin. It encodes inside of the beta cells of the islets of Langerhans, which is found in the pancreas of animals. And like I talked about, it contains 81 total amino acids. With type 1 or type 2 diabetes, the beta cells are actually either destroyed or they're actually damaged. And what this does is this prevents insulin from actually going to the glucose to perform what it needs to do. And it can cause the damaging effects of hypoglycemia, which can be impaired sensory perception, like when there is blurry vision. You can actually start to get dizzy. You can go into a coma and actually encounter death. So the importance of insulin cannot be stressed enough, with the fact that the amount of people projected to have type 2 diabetes in the year 2025 is 300 million people, almost triple the amount in the year 2000 with 120 million. If it wasn't for insulin, it'd be a lot more difficult for organisms to circulate energy throughout their body structures, and more energy would be wasted maintaining their metabolisms.